Glycogen branching enzyme. So as the glycogen synthase continue to produce the alpha 1 4 chain of glycogen, the linear chain is going to get longer, and at some point the alpha 1 6 branches has to be created from somewhere. So this enzyme kicks in this glycogen branching enzyme. So as you get a certain up to the certain length of the glycogen chain, these glycogen branching enzymes will hydrolyze alpha 1 4 of the glycogen chain and reattach it to the as the alpha 1 6 branch to the previous chain. Okay, so thereby producing two non-reducing end, and the process repeat. So it's the collaboration between glycogen synthase and the glycogen branching enzyme. So in the end, you're gonna get the molecules of glycogen with so many branches, and this is to help with the speed of glycogen breakdown when the cells need to do so. Now, regulation of glycogen synthesis and glycogen breakdown. There are two important enzymes in the process. One is glycogen synthase and the other one is glycogen phosphorylase. Okay, you need to understand that these two enzymes are regulated in a similar fashion but somewhat opposite. For glycogen synthase can be phosphorylated, and the result from phosphorylation yield the inactive form of the enzymes. We call it a B form. Okay, whereas for glycogen phosphorylase, the phosphorylation of glycogen phosphorylase use the active form or the A form. So this is a cartoon trying to illustrate the phosphorylation of glycogen phosphorylase. So we start from the two subunit of glycogen phosphorylase. This enzyme is an allosteric enzyme, by the way. So it has the serine side chain, serine number 14 at the side chain of it. Okay, and under the condition that the body releases glucagon or epinephrine. Okay, so this signal is gonna promote the activity of this enzyme phospholase B kinase. So phospholase B kinase will phosphorylate the serine residue of glycogen phosphorylase to make this an active version of the enzyme. Okay, and when this active form is not needed, the phospholase A phosphatase, or we call it in short PP1, is going to dephosphorylate the enzyme to make it in an inactive form. Okay, so this PP1 is activated by insulin or glucose 6-phosphate and glucose. This is a cartoon showing the cascade of glycogen phosphorylase activation. It is divided into two cell types. One is the myocyte or mast cells, and the other one is hepatocyte. Okay, so muscle cells can can receive the signal from epinephrine. Okay, but actually epinephrine can activate both mast cells and the liver cells. But for the liver cells, it can only receive the signal from the glucagon, and the glucagon is gonna, so upon the activation, the G protein is activated. The receptor for this enzyme is gonna activate the G protein. The G protein will then activate the enzyme adenylocyclase. Adenylocyclase is the enzyme that responsible for making cyclic AMP. So the cyclic AMP will in turn activate the enzyme protein kinase A or PKA. Okay, the enzyme protein kinase A will activate phospholase B kinase. So this phospholipase B kinase, the activated one, the active one, will phosphorylate glycogen phospholipase. Okay, so glycogen phospholipase will become active, and the active glycogen phospholipase will hydrolyze glycogen into glucose 1-phosphate. As a result, glucose 1-phosphate enters muscle cells and it will go to glycolysis to make energy. But for the liver, it will eventually be converted to glucose, and the glucose is sent back to the bloodstream. Okay, so you are gonna need to remember or get familiar with this cascade of glycogen activation. Let me skip this. I don't think this is necessary for you, so I'll just skip it. I'm not gonna talk about it. Okay. The control of glycogen breakdown in the liver. So the liver version has the similar enzymes to the muscles, but they are different in some of the composition. They are not produced from the same gene, but this is what we call the isosan. So the similar genes produce similar enzymes but regulated differently. So in the liver, the active form of glycogen phosphorylase, the side chain of serine 14, is hidden, and then when the glucose is being produced by glycogen breakdown, so the glucose is accumulated, the glucose is gonna come back to the glycogen phosphorylase and bind to the allosteric site, thereby inducing conformational changes to the enzyme. So there is an exposure of the side chain of serine 14. So this phosphate group is exposed, and then this is an easy target for phospholipase A phosphatase, so PP1, which is activated by insulin. So phospholipase A phosphatase will cut down the phosphate group to make this enzyme less active. Okay, so it's the relationship between glucagon and insulin. When the body is starved, the body will produce glucagon or epinephrine to stimulate the glycogen breakdown to make more glucose. But as glucose is accumulated, insulin will in turn get in action. So insulin will tell the body that, hey, there is now so much glucose, we need to stop producing glucose. So the insulin is gonna stop the action of phospholipase through PP1 enzymes, so then stopping the breakdown of glycogen but to make more glycogen. Okay, so this is for the action of glycogen synthase. Glycogen synthase is active only when there is no phosphate group on it. So the enzyme that phosphorylate glycogen synthase is called GSK3, glycogen synthase kinase 3. The GSK3 will put the phosphate group to the glycogen synthase to make this enzyme inactive. Okay, so the signal from insulin will inhibit the action of this enzyme, and that makes sense because when you have insulin, it means you have a lot of glucose, and so you want to continue to make glycogen. You want to pull the glucose out of the bloodstream. Okay, but in contrast, when the enzymes, okay, so insulin activates PP1 and then inhibit GSK3. Okay, but if there is a signal from glucagon or epinephrine, these two hormones will inhibit the PP1 from taking away the phosphate group. Okay, just so that the glycogen phosphorylase will be active. Okay, so this is the interrelationship between the process of glycogen production and glycogen breakdown. what I have here on the lecture might not be crystal clear to you, and so I highly encourage you to go to the textbook and do some additional readings just so that you gain more understanding about the regulation of glycogen synthesis and glycogen breakdown. And in fact, this enzyme is actually a really important enzyme because the action of this enzyme controls both glycogen synthesis and glycogen breakdown. So it's kind of like a master of regulator for um, metabolism of glycogen. And that's it for metabolism of carbohydrates. And I hope you do your own additional readings on top of my lecture. Okay.